This is a product video demonstration of the Opiates 865 6-panel saliva drug test. In front of you are the component parts of that test as supplied for a single test. This test comes packed in cartons of 15 from the manufacturer with a full data sheet. The panel itself is foil wrapped, has standard in vitro diagnostic markings on the back, including the 6 drugs that are included in this test, the expiry date and the lot number. The oral fluid collector or sponge on a stick is separately foil wrapped, as is the saliva or oral fluid chamber, collection chamber. So let's take these out of their wrappings and show you the component elements of this test. Here we have the panel drug test. This is a 3-column panel test with 3 sample wells. Control lines marked by C on the cassette and the T lines marked with their abbreviations at the head of each column. So if we look at this left-hand column, you have T1, T2 and T3 corresponding T1 to cocaine, T2 to amphetamine and T3 to benzo. The swab on a stick comes out of its packet with the sponge absolutely firm and rigid. This is an inert collection sponge on a stick which is given to the donor to saturate with saliva. When it is saturated, the swab becomes soft and fluid. This is the collection chamber for the saliva. It unscrews into 2 elements. This is the chamber into which the sponge is squeezed and discharged into this bulb at the end, and that little arrow, the black arrow, is the minimum saliva that you are aiming to collect. So that is the sample. There are markings on here which are very difficult to see on a video but correspond to equivalent mils. When that is filled with the saliva, the screw lid is reapplied. You can mark this with date and initials from the donor, and then by flipping this little pipette device on the top, you are then able to invert it and dispatch saliva into the sample wells to run the test. So what we're going to do now is demonstrate that happening. We're going to saturate the sample collecting swab with saliva and run a test and show you how that functions. Now this swab has been completely saturated with saliva. Expect saliva collection to take between 2 and 5 minutes. When saturated with saliva, it's soft and quite heavy. It's critical that the swab, once softened, is worked well around the oral cavity to collect any cannabis deposits. But when you're ready to run the test, the saturated sponge is simply pushed into the base, and you can see the saliva dispatching into the bulb at the base here, and that is just over the minimum that we require. Can push quite firmly to dispatch all the saliva from the swab, then that can be disposed of. Place the lid on the saliva collector, flip up the little pipette and invert the collector, and then squeezing gently dispatch 3 drops of saliva into each of the sample wells, and then start a timer for between 5 and 10 minutes. Follow the manufacturer's guideline for the correct read time, but you can already see the pink dye starting to appear at the base of your results columns, and you now need to leave this for the desired time, and then we will show you the results and how to interpret the results when the read time is complete. Well, this test membrane has now been running for 5 minutes. The first thing to check is that you've got good quality control lines marked by the C on this cassette in all 3 columns. They're clearly evident in all 3 here. The pink dye is just starting to clear now and will clear further by 10 minutes. But you are then looking for the presence or absence of T lines or test lines for each of the drug groups corresponding to the abbreviation. And you can clearly see here in the left-hand column, you've got 3 really strong T lines indicating negative results for cocaine, amphetamine and benzos. In the middle column, you've got 2 clear test lines, T1 and T2, corresponding to negative results for opiates and methamphetamine. And in the right-hand column, you've got a faint T line for the cannabis test. Now, that is perfectly normal. You must not interpret anything by the colour density, thickness or saturation of the test line. That is a clear negative result for cannabis. Cannabis membranes test down to 20 nanograms, and in some cases with the new modern tests even lower. So these are very, very sensitive drug tests, and the test lines tend to be much thinner and fainter than the corresponding other drug groups. So here we have a clear negative result for all 6 drugs, with control lines indicating that all those test results are valid. If none of the test lines were showing or one was absent, you would need to wait for the full 10 minutes development time and read the results showing at 10 minutes for this test. If no test line is developed at 10 minutes in the presence of a control line, that would be a non-negative, possibly positive result, and the sample would need to be sent to a laboratory for further analysis to determine the cause of that non-negative result. So there we have the process, parts, and how to interpret the results on an all-test DSD-865 6-panel saliva drug test. Thanks for watching.